In this video, I would be talking about G-protein coupled receptors. Overview of this presentation will include what are G-protein coupled receptors with examples, how does G-protein function, the various targets of G-proteins, types of G-proteins and the signaling pathways with various examples. So to start off with G-protein coupled receptors, what are G-protein coupled receptors? G-protein coupled receptors are also known as metabotropic receptors or 7-transmembrane spanning receptors. The structure comprises of 7 transmembrane helices with an extracellular N-terminal and intracellular C-terminal. They are membrane receptors that are coupled to intracellular effector system via a G protein. The G protein consists of subunits such as alpha, beta, gamma, which exists as a trimer anchored to the membrane through lipid residues. They constitute the largest family and includes receptors for many hormones and slow transmitters. For example, the muscarinic acetylcholine receptor, adrenergic receptors, and chemokine receptors are some examples. So what is the role of G proteins? G proteins comprise a family of membrane resident proteins and consist of alpha, beta and gamma, which exist as a trimer and reside in the membrane. The function is to recognize activated G protein coupled receptors and pass on the message to the effector system that generates a cellular response. So next is, how do G proteins function? The G proteins function in 4 stages. The first one is the resting state of the receptor. The second is the activation of the receptor. Third is activation of target proteins. And fourth, termination of signaling and return of the receptor to the resting state. So initially the receptor is in the resting state. It is functionally silent in the absence of ligand binding. Alpha, beta, gamma subunits of G protein exist as a trimer and reside in the membrane with GDP attached to alpha subunit. The target proteins are inactive. So this is the resting state of the receptor. So the next state is receptor activation. Here the ligand binds to the receptor and causes a conformation change in the receptor. The conformation change in the receptor helps the receptor to acquire affinity for alpha subunit of G protein. The alpha subunit with the GDP attached to it moves towards the receptors and binds to the receptor. Subsequently, the GDP molecule is replaced by GTP and the receptor is activated. This helps in the generation of signal. So the next step is activation of target proteins, which includes enzymes and ion channels. The alpha GTP complex dissociates from the receptor, moves towards the target and activates the target proteins. This initiates and generates a signal. It activates the signaling pathways and generates a cellular response. The beta gamma subunit of the G protein also moves towards the second target and activates the target protein. The next step is termination of signal. Termination of signal is brought about by hydrolysis of GTP molecule into GDP and inorganic phosphate. The alpha GDP complex now dissociates from the target protein and reunites with beta gamma complex of G protein and returns to the resting state. So what are the targets for G proteins? The main targets for G proteins through which G protein coupled receptors control different aspects of cell function are adenylyl cyclase, the enzyme responsible for cyclic AMP formation, phospholipase C, the enzyme responsible for IP3 and DAG formation, ion channels, particularly calcium and potassium channels. Many different drugs, hormones and neurotransmitters act on G protein coupled receptors and produce the effects by increasing or decreasing the catalytic activity of adenylyl cyclase, thus raising or lowering the concentration of cyclic AMP within the cell. The various subtypes of G proteins are GS, GI and GQ. GS is stimulatory G protein and GI is inhibitory G protein. The G protein coupled receptors acting through GS protein increases cyclic AMP formation, which activates protein kinase enzyme which controls phosphorylation and regulates various cellular functions. For example, in the heart muscle, phosphorylation caused by protein kinase enzyme increases the activity of voltage activated calcium channels. This increases or enhances the calcium entry into the cardiac cell, increasing the force of contraction and increasing the heart rate, force of contraction and conductivity. In the smooth muscle cells, cyclic AMP dependent protein kinase phosphorylates myosin light chain kinase enzyme which is required for contraction. Phosphorylation of myosin light chain kinase inactivates the enzyme and this inhibits contraction, and this accounts for relaxation of smooth muscle cells. In the next example, increased cyclic AMP production in response to beta adrenoceptor activation affects enzymes involved in glycogen and fat metabolism in the liver. So, activation of beta receptor increases cyclic AMP formation, increasing the phosphorylation which is caused by protein kinase A. Phosphorylation activates the enzyme, that is phosphorylase enzyme, which is responsible for the breakdown of glycogen to glucose, and glucose is utilized by the cells. The next subtype of G protein is G inhibitory protein. Examples of G protein coupled receptors acting through G inhibitory protein are opioid receptor, alpha 2 adrenergic receptors, M2 receptors which are present in the heart. The opioid receptors are linked to G inhibitory protein. Activation of opioid receptor decreases cyclic AMP formation which leads to activation of potassium channels. Opening of potassium channels causes efflux of potassium ions causing hyperpolarization and decreasing the excitability in the neurons. The next example is alpha 2 adrenergic receptors which are located in the presynaptic nerve endings of the adrenergic nerve terminals. So alpha 2 receptors are inhibitory in nature and form a part of feedback inhibitory loop. That is, they inhibit the release of adrenaline and noradrenaline from the adrenergic nerve ending, thus decreasing the sympathetic neurotransmission. The third example is that of M2 muscarinic receptors which are present in the heart. M2 receptor is a G protein coupled receptor linked to GI protein, decreases the cyclic AMP formation which decreases the calcium conductance due to decreased phosphorylation, and decreased calcium conductance decreases the cardiac contractility, heart rate and conductivity in the heart. The next subtype is GQ protein. Activation of GQ protein stimulates the formation of IP3-DAG pathway. This catalyzes the formation of IP3 and DAG. IP3 acts to increase free cytosolic calcium by increasing the release of calcium from intracellular compartments, increasing free calcium and initiates many events including contraction and secretion. DAG activates protein kinase C which controls many cellular functions by phosphorylating a variety of proteins. Examples of receptors that act through GQ protein are alpha 1 receptors which are responsible for smooth muscle contraction. Alpha 1 receptors are present in the smooth muscles of the blood vessels and activation of these receptors causes vasoconstriction. Alpha 1 receptors are also present in the neck of the bladder, prostate, which causes constriction of the urinary sphincter and may cause urinary retention. Alpha 1 receptors are also present in the radial muscle of the iris. So, stimulation of alpha 1 receptors contracts iris, causing dilatation of the pupil. The next receptor is M3 receptors which are present in the smooth muscles of the bronchi and urinary bladder. Stimulation of M3 receptors causes contraction of bronchi, causing bronchoconstriction and also contraction of urinary bladder. M1 receptor is linked to GQ protein and is responsible for salivary gland secretion and secretion of gastric juice. This brings us to the end of the topic. These are my references. Thanks for watching. Please like, subscribe and share.